Our next presenter is Justine Morales from the Southern Methodist University.
Hello, everybody. Hello, everybody. My name is Justine Morales. I am a senior at Southern Methodist University. I'm a biology major, Spanish minor, originally from Stratford, New Jersey. And this previous summer, I got to work with Dr. Helen Lay and Dr. Jane Johnson at UT Southwestern in the Department of Neuroscience Neuronal Developmental Lab on a project entitled A-to-1 Lineage Neurons Define Non-Canonical Subsets of Cerebellar Projecting Neurons, critical for coordinating motor tasks. So this all started back in the 1830s with Sir Charles Bell. He came up with this quote, "When a blind man or a man with eyes shut stands up straight, by what means is it that he maintains an erect position? Is it obvious that he has a sense by which he knows the inclination of his body and that he has a ready aptitude to adjust it? I call this the sixth sense." And this idea of a sixth sense was further brought upon by Sir Charles Sherrington in the 1900s, where he was studying reciprocative inhibition circuits. So your ability to contract your bicep is caused by your ability of your tricep to relax and become inhibited. So he was cutting different muscles, different neurons, and he actually ended up cutting what is known now as one of the afferent proprioceptive neurons. And when this happened, if you'll play this video, you see on the left there's a wild-type mouse, and on the right there's a mouse with its proprioceptive afferent. Oh, I guess the video doesn't work. Proprioceptive afferent neurons cut off, and actually, I can show you anyone who wants to see this video. I have it on my laptop. But basically, the mouse can't sense where his limbs are, so in order for it to walk, it has to kind of pull it directly to its body and then fall on it. And then it extends it again and does that kind of motion in order to walk rather than reaching and being able to actually use its limbs to their full ability. And this is also seen in humans. This is supposed to be another video, but this man actually suffers from an autoimmune disease where his immune system attacks his proprioceptive neurons. So he actually cannot sense where his body is relative to itself, but he managed to teach himself how to walk, and there's this whole documentary about him. And basically it's because when he walks, he has to physically look at his legs to walk so that he can see where his legs are so that he can move them accordingly. So if there's ever a blackout or for some reason this man is blinded, he'll automatically fall to the ground because he can't sense where he is. So this is the concept that my lab was working on. So based on some previous research that was done, we know that A-to-1 lineage neurons go on to create proprioceptive neurons. And A-to-1 is a group of the basic helix, loop helix transcription factor family, which is found in the dorsal part of the neural tube. So these go on to make your proprioceptive afferent neurons. And what my lab was trying to discover is where these neurons are located, whether in the brain, where they go in the brain, and whether they go to specific muscle groups, and from there extend to other muscle groups, or whether there are kind of proprioceptive stations that can help you monitor your body. So to do this, we had to create a tracer mouse. So we crossed a Cree knock-in mouse with a reporter mouse. So the Cree knock-in mouse has the Cree recombinase following the A-to-1 locus, and Cree recombinase is able to flox out LOXP1 sites. So these LOXP1 sites were put around the stop codon following TD tomato, which is just a basic fluorescent that is genetically implanted into the reporter mouse. So these LOXP1 sites go on both sides of the stop codon so that when they're crossed, the Cree recombinase will actually cut at the LOXP sites and remove the stop codon, allowing TD tomato to be expressed wherever A-to-1 is. So now after this cross, it's preceding offspring, we can see where A-to-1 lineage neurons are and we can actually track them. And so we did this TD tomato is this kind of purplish color here, and Nguyen is actually used just to label the general population of neurons. So this is a spinal column in the thoracic area, and what we did was we wanted to see if we could find any general co-localization between neurons and ATO1 lineage neurons, and we found actually a couple sites. These medial and lateral areas are kind of already known and talked about generally, but these motor neurons and sympathetic preganglic neurons are actually, it was pretty astonishing to see these because when you look at the neural tube, ATO1 is derived up here in the more dorsal area, while OLIC2 is found in the ventral area, and OLIC2 is actually what goes on to create your motor neurons. So we began to question, if ATO1 is in the dorsal area and OLIC2 is in the ventral area, how is it that these two different progenerative neurons go on to create potentially the same neuron down the line? So we wanted to confirm that this co-localization was in fact with motor neurons, so we did another immunostain with HB9, which stains motor neurons, and we found that in specific areas of the spinal cord, like the upper cervical, lower cervical, upper lumbar, and lower lumbar, there was actually co-localization in specific regions that are known to be associated with the phrenic region, or it's part of your diaphragm, the forearm flexor, the psoas, which is part of your hip flexor, and the foot. So from these general areas, we had ideas of potential muscle areas that might be working to help you get that proprioceptive sense. And so we had a couple of predictions. What I basically did my summer, last summer, was this is kind of an abstract picture, but this is supposed to be a mouse's head. Its nose is up here, body is down here, and you can see that there's a needle there. So what we did was, because the TD tomato was attached to ATO1, the cerebellum and the brain of the actual mouse would fluoresce, which you can partially see here in the slight red. So it would fluoresce, and what we did was we injected CTB, or chlorotoxin B. It's a retrograde neuronal dye. So what we did was we injected that into the cerebellum to see if it would be traced down into the spinal cord to potentially find other motor regions that it may affect. So with these are just some pictures of different injections that I've done on the cerebellum. I found that sometimes it's actually pretty difficult to do things of that nature because you're sitting there looking at a mouse and you have to figure out, based off the morphology of its head, where its cerebellum is. So there were a couple of times where I may have been off slightly, but I did get a couple of good regions where I was able to actually hit the cerebellum, which was pretty exciting. But unfortunately, we weren't able to find any co-localization in the actual spinal cord. But what we also did, other than injecting solely in the cerebellum, was we injected in the muscle groups themselves that we found correlated with the spinal groups. So that would be the bicep, tricep, quadricep, hamstring, and the foot. And we actually did get a couple of successful retrograde hits. So when we injected the CTB in the spinal cord, we got a couple traces that actually came back into the cervical area of the spinal cord, which is related, or where you would expect to see neurons projecting from the bicep region. But unfortunately, as you can see, these are the TD tomato cells highlighted in red at the top, and these are where the CTB labeled. So these two groups were actually distinguishable. They weren't co-localized in any manner. So this, although it did actually trace back, did not kind of go with our hypothesis. And the same is seen here in the hamstring that we did. Again, this is the spinal cord. This is the lower lumbar region. But there is a very minimal co-localization, so it wasn't enough for us to say that we actually did find some regions that may be proprioceptive stations. So again, just the injections were promising. They were slightly difficult to do, but at the end of the day, we were able to do them. And we also, in addition to that, we wanted to make sure that any of these changes that were caused by the knockdown of ATO1 were only caused by the knockdown of ATO1 and not any additional side effects or factors. So I helped my PIs create this picture, which is actually published in the article. And so what we did was we did a HOX4 Cree ATO1 cross. So that A-1 was actually only knocked out caudally, and you can see it's still died here, because if you knocked out A-1 completely, it's actually lethal, so you come out with stillbirths, and that's obviously what we're trying to avoid. So, and when we're speaking on the effects of proprioception, this is another video. This is a wild type, and this is supposed to be a A-1 knockout, and what they do is they're just simply supposed to cross a ladder. Obviously there's nothing, well, there is something under the ladder, but there's space for the mice to actually miss. So the wild type would just, casually walk across the ladder, no problem. But the mouse that had its proprioceptive neurons knocked out actually missed almost every other ladder rung. You'd see it basically almost fall through. So we want to make sure that those are only caused by the knockdown of A-1. And so what we did was we did some dyes. So we took pre-birth neonatal pups about 10 and a half weeks in. And here in the wild type, you see A-1 is labeled here, but it is in fact knocked out in the conditional knockout. And we also want to look at LHX29, which is a downstream differentiated form of A-1. So here you can see it labeled in the wild type, and again, it's knocked out in the conditional knockout. So that was a positive reinforcer. And we also want to make sure that there were no effects to any motor neurons. So we did HB9 in between wild type and conditional knockout, found there was no true statistical difference between the two. And finally, we just stained for actual proprioceptive afferent neurons. And again, there was slight variance in a couple groups, but there was no true statistical difference between the two. So we were able to confirm that any and all changes that were seen between the wild type and the conditional knockout were actually caused by the deficit in the A-1 locus. So that actually wraps up what I was able to do last summer. This is the Jane Johnson lab that I got to work in. So I want to acknowledge these people in PSCP specifically. If there are any questions, I'd be willing to answer them. Yes.
Is it known what subset of genes it works on? Or is that not in here, what the target genes are?
That was actually someone else in the lab was actually working on that, so I haven't, I don't know personally. Thank you.